This is the Viral Pathogenesis video series. This is going to be a multi-part video, and in this portion we are going to be talking about attachment, invasion, and evasion tactics that are associated with viral pathogenesis. When discussing these aspects of viral pathogenesis, it's important that we are talking about animal viruses, which are going to affect eukaryotic cells. In other words, they are going to infect our own body. So these are the animal viruses, or the types of viruses that would cause viral pathogenesis within us as humans. So the first thing is that we have attachment. Just as bacteria attached, viruses have to attach as well. So viruses attach through their spikes. Remember that viral spikes are going to give the range in which a virus can infect a certain cell. So the spikes will infect or attach to a certain receptor. So if that receptor is not on a cell, then therefore that virus cannot infect that cell. So we're going to say attaches to specific receptors. And then you're going to get invasion. Invasion with viruses are just going to be the methods through which viruses enter into a cell. And the two ways through viruses enter are through enveloped fusion and endocytosis. Enveloped fusion is commonly known as just fusion. And the reason I wrote enveloped fusion is because it reminds you that in order to do fusion, you have to have an envelope, which is that lipid membrane structure that's obtained through budding. So these are the two ways through which viruses invade. Once they invade, they're going to be able to replicate in the cytoplasm or replicate in the nucleus, but the viruses are going to replicate inside the cell. Then we have evasion. Remember that evasion is to evade or avoid the immune system. There are several different techniques through which viruses can evade the immune system. The first way is through the envelope. The envelope is obtained through our cells. Viruses obtain their envelope as they leave a host cell. Therefore, this lipid membrane is going to appear as our host cell. So this avoids detection by the white blood cells. And the way it avoids detection by the white blood cells is because an envelope acts like camouflage. Another method through which viruses can evade is through antigenic variation. Bacteria also can undergo antigenic variation. So remember that antigen is anything foreign to the body. Variation means that it changes. So this is changing how it looks. So the body can no longer have memory against it, and new memory has to be created. However, with viruses, we're going to have two different terms that are associated with antigenic variation. That is genetic shift and genetic drift. So genetic shift, when you think you're shifting, you're moving somewhere quickly. So genetic shift is going to be a quick change, and this is also known as genetic reassortment. Genetic drift, if you think of something drifting, or like a piece of wood drifting in a river, it's doing it very slowly. So genetic drift is a slow change, And it's because there's mutations that are building up over time. I want to spend some time now on genetic reassortment and explaining what's occurring here. Genetic reassortment is when two viruses are going to affect the same cell simultaneously. Both genetic shift and drift are commonly seen with influenza. So let's say I have a cell here and have two viruses, two strains, I should make sure to say, of the same virus. So this would be two different types of influenza virus. So they're the same virus, but they're different strains. So they're just different a little bit in structure. So I have virus A, which is going to be in red. And then I have virus B, which is in blue. So these viruses are both going to infect the same cell at the same time. And when they get in there, they're going to replicate. So we're going to have more genomes, more spikes, and more capsids for the blue one. And the same thing with the red. You're going to get more genome, more spikes, and more capsids. One thing that should be made noted is the fact that my genomes are in pieces. This is key for genetic reassortment to occur. In order to have genetic reassortment, your genome has to be in pieces, which is what we call segmented genome. So in order to do genetic reassortment, you have to have a segmented genome. And that's because these pieces are now going to interact. So the red and the blue are going to form a new virus, a new strain. Let's say it has a blue capsid, half blue genome, half red genome, and red spikes. This virus right here does not look like this one, nor does it completely look like this one. Therefore, this is a new strain. So this was the genetic shift. So it happened when they both infected the same cell simultaneously, the pieces interacted, they reassorted themselves and created a new strain. Now this new strain can go infect a new cell and it will replicate its own thing and start making more and more of this new virus. So this was a very quick process, while genetic drift are mutations that build up over time. The last evasion tactic is going to be the fact that viruses can be latent. So I'm going to draw it over here. So viruses can have an envelope, antigenic variation, or they can go latent. So when a virus goes latent, it's hiding within the body. Usually it hides within the nervous system, within nerve cells. But what's occurring is the fact that the virus is staying inside a cell, but it's not replicating. So the body thinks it eliminated it, but the virus is just hiding for a long period of time. So the goal of latency, or to go latent, is the fact that you hide and you don't reproduce in order to avoid complete destruction by the immune system. Eventually you can reoccur and come back and make more of your cells. A great example of this is the herpes virus, herpes simplex 1, which causes cold sores. Many people that suffer from cold sores do not get them just once. They'll get it, it'll go away for a couple months. Then it comes back, then it goes away. And then it comes back, and so forth. So that is what happens with latency, is the virus comes back, shows itself, goes back in hiding to avoid complete destruction, then comes back, then hides again. So that's a wonderful tactic that viruses have as an evasion tactic, because they completely evade the immune system from destroying them. So these are your attachment, your invasion, and your evasion techniques that are associated with viruses.